Okay, how's it going, Alex? Hope you are doing okay through all of this. This is a little weird, but let's see if this works. I'm going to just explain it, and hopefully, I'll cover it in a way that it makes sense. But if you have any other questions, obviously, just shoot me another email. But hopefully, this will clear some stuff up. Okay, so it seems like the disconnect is you have no problem getting the Punnett square filled in. Hopefully, you understand the idea of the homozygous dominant, heterozygous, homozygous recessive. But I'm just going to go over that with you real quick. And then I'm going to make sure you understand this idea of a ratio and filling in these ratios. Okay, so all this is, it's a Punnett square is a tool, like I was saying in the lecture, and it's a way to look at the probable outcomes of a cross. And you can use it for anything. It doesn't have to be genetics. You can use this if I want to see my possible outcomes of, taking two coins and flipping them, like I said in that example. So, in the case of this problem, this is a dominance problem. So, big T is dominant to little t, which means that tall plants are dominant to short plants, and you are going to have three possible options of alleles in this scenario. You could have two big T's, right? That's one possibility. The next possibility is you could have a big T and a little t. You could also have a little t and a big T. But since that's the same thing, we just kind of consider it to be one. So it's big T, little t, big T. And then the third possibility is you could have two little t's. So if we're just looking at possibilities in general, there's a 25% chance that you're going to have two big T's because there's four possibilities, right? Because this one is big T, and little t, big T. So that would be 25 and 25. So that'd be a 50% chance. And then finally, you would have a 25% chance of just getting two little t's. So what that means is overall, you have a better chance of getting a big T and a little t than either of the other two. So let's talk a little more about this idea of dominance, right? So dominant means that the big T overpowers little t. So I like to think of like a shoe. It's hard to draw with this thing, but imagine a shoe from the big T crushing the little t. It just overpowers it. So in this scenario, you have a better chance of having big T, little t, or big T, big T. So that's why most plants are going to be tall, because it's more common to have that combination. The only plants that are going to be short are going to be the two that have little t's. So let's just do this Punnett square real quick and see if it makes some sense. Okay, so it says, what is the genotypic and phenotypic ratio of a cross between a heterozygous tall plant and a short plant? So hetero means different, so this would be heterozygous tall, right? Because it'd be big T, little t. Hetero means different. And then a short plant. Well, short is going to be recessive, so the only way to have a short plant is for it to be little t, little t. So these are the two plants. And I want to know what is going to be the genotypic and phenotypic ratio of a cross between these two. So we're going to put one parent on one side, and we're going to put the other parent on the other side. Now, again, it doesn't matter. I could have done it the opposite way, as long as you group the alleles together for each parent. So I could have put little t up here and big T, little t down here, but I didn't. Okay, so we're going to fill in the Punnett square, and you said you understand that, right? So we're just going to do big T here and little t from here. This one's going to be two little t's. This one is going to be big T, little t. And then this one is going to be little t, little, oh, to make another one. So that is the possible outcomes of a cross between those two individuals. Now I want to know what are the ratios, or I could have said, what's the chance of having a short plant? What's the percentage chance? So the way that's done is we have four options, right? So it's 1, 2, 3, 4, which means everything is going to be either 0, 25, 50, 75, or 100%. So just to make sure we understand genotype versus phenotype, the genotype refers to the actual genes, the actual genetic code. So that refers to the letters, the alleles. The phenotype is the physical outcome of that genetic trait. So for example, if I were to go into your DNA and I were to analyze it, I would find that you have the genetic code for brown hair. So your genotype would be the actual genetic code in your DNA, and then the phenotype would be the fact that you have brown hair. So if I were to go in and alter it, and let's say I were to change your DNA around and I would make it so you have blonde hair, I would do that by changing your genotype. So I would actually go in and change your genetic code, and then that would end up giving you blonde hair as the phenotype. So the genotypic ratio, all a ratio is a way to compare something to something else, right? So for example, if I said, what's the ratio of boys to girls in your house right now? You would count up the number of guys, so you and your dad or whoever, and then you would count up the number of girls, and you would say, all right, there's a 2 to 3 ratio guys to girls, which means that there's 2 guys for every 3 girls. So we're going to do the same thing here, but we're looking at the genotypic ratio. And the way that's written is it always goes like this. Homozygous dominant goes first, heterozygous goes in the middle, homozygous recessive goes at the end. So we're going to count them up. Oops. Come on. Sorry. All right, so we're going to count them up. So let's see. How many of them are going to be big T, big T? Well, if I look over here, I see that there's none. There's 0. So we're going to write a 0. How many of them are big T, little t? Because that's hetero. Well, here's 1. That's not 1. Here's 2. That's not 1. So we know there are 2. And then finally, homozygous recessive is little t, little t. So how many of them are little t, little t? Well, we already counted those. So we got 1, 2, 2. So it's a 0 to 2 to 2 ratio. Now, phenotypic ratio is I want to know dominant to recessive. So how many of these are dominant? Well, as we know, big T is dominant to little t. So these two are going to be tall. So we're going to have 2 tall. And then here we have 2 that are little t, so there's going to be 2 that are recessive. So that is going to be a 2 to 2 phenotypic ratio. Now let's say I want to know what percentage chance do I have of having a short plant. Well, if there are 4 options and 2 of them are short, that would be a 4 to 2 or a 50% chance, right? 50%. If I said, what are the chances of having a homozygous dominant plant, like a tall plant that's big T, the answer would be 0% because none of the options are that. So if it was 1 square, it would be a 25% chance. If it was 2 squares, it would be a 50% chance. 3 squares is a 75% chance. All 4 squares is a 100% chance. And then no squares is a 0%. So let's try another one out just to see if it makes sense to you. Let's say I want to know the same question, right? But instead of it being a heterozygous and a short plant, I want to know 2 heterozygous plants. So instead of it being big T crossed with big T, little t, we're going to do big T crossed with big T. So let's say both plants are going to be heterozygous. So I'm going to fill in my Punnett square, big T, big T, little t. Now this might be overkill. You might already have got like, oh, this makes sense. So, you know, but I would still just watch this example just to make sure you get it. And then hopefully this makes sense and this is what you were trying to figure out. If not, email me and we can do this again. I got all the time in the world. Okay, so let's do the ratios. So in this scenario, I want to know my genotypic ratio. How many of them are homozygous dominant? Well, if I come over here, now it's a different number, right? So it's big T. There's 1 of them, so it's a 1. How many of them are heterozygous? Well, I have 1, 2. And how many of them are homozygous recessive? The answer would be 1, right? Because there's just the 1 little t. This number should add up to 4. So if you could have just looked and said there was only 1 left, so you could have figured that out that way as well. And now I want to know the phenotypic. Oops. Now I want to know the phenotypic ratio. Oh man, I'm making a mess out of this. We'll just leave that crossed out. Okay, the phenotypic ratio is dominant to recessive. So how many of them are dominant? Well, we have 1, 2, 3, because big T is dominant to little t. So both the homozygous dominant and heterozygous genotype are going to be big T, be tall. And then how many of them are recessive? Well, just this one here. So let's say the question said, what's the chance of 2 heterozygous tall plants producing a short plant? The answer would be 1 out of 3 would be 25% chance. If I said, what are the chances of 2 heterozygous tall plants producing a tall plant? The answer would be 3 out of 4, which would be 75%. So hopefully that makes sense. Please watch this right away. If you